Welcome back to BioMinds, your guide to mastering molecular biology and launching a high growth career in the life sciences. If you're new here, make sure to hit that subscribe button so you never miss a lecture. Let's get started. If you wanted to go from home to some place, okay, so what you will prefer? You will use some transport, right? So, why you use transport for changing the location? So this transport will travel or transport you from one location to another easily. There is a less you will have the strength. Okay, means if you are walking from one location to another, so you might lose some strength, right? But if you use vehicle, so it will be easy. There will be less consumption of your strength, and you will reach the location safely. Okay, so we use certain vehicles. So there are so many vehicles that we use for changing the location. So just like that, suppose if I wanted to transfer some genes from one cell to another, so what I will use? Okay, because gene is very critical product of the cell. So if you just take out the gene and just transfer it directly to the another cell, so there are chances of damage. Okay, you will not transfer it easily, as well as there will be damage to this genes. There is a loss of sequence. Okay, so to reduce that, you should have something, right? So how you can do that? How can you transfer gene from one cell to another without damaging it and easily? So the answer is vector. Okay, so vector is just like your vehicle that helps you to transport from one location to another. So vector is the vehicle that transport the gene from one cell to another, or from cell to the lab, or to the different host cells. Okay, so today we will discuss about vectors. So there are so many different vectors available. Okay, and this vectors has very important application in the recombinant DNA technology, or you can say genetic engineering. So before this, I have already uploaded a lectures on genetic engineering introduction, as well as the enzymes used in genetic engineering. So you must watch those videos first. So if you understand, if you know these enzymes, cut the DNA and paste the DNA into the new location, so the vectors comes into picture. So vectors helps that new DNA to transport from one cell to another. So today we will discuss about the vectors. So there are so many different vectors we have. So the DNA, the vector is nothing but DNA molecule. It's also a DNA sequence, okay, that is used as a vehicle. I have already mentioned. Okay, from host cell to the cloning or the recombinant DNA technique. Okay, so it is naturally occur. Okay, it is present in the certain bacteria. Okay, it is a naturally DNA molecule which is present in certain bacteria, bacteriophages, and viruses. Okay, the very first vector was the plasmid vector, was isolated from the bacteria. So today we will discuss about plasmid vector. So there are certain types of vector. Here you can see the plasmid vector, viral vector, which is derived from the virus, cosmid, phage vector, bacteriophage, artificial chromosome, which is modified version of the vectors. So today we will just discuss about the plasmid vector, because it is very important and it is first discovered vector. So the plasmid vector, as you see, if you remember, the plasmid is the natural gene or natural chromosome present in the bacteria, certain prokaryotic cells. So suppose this is the cells of the bacteria. So here they have their own DNA and they have some extra DNA which is circular. Okay, and that DNA is known as the plasmid. Okay, so this plasmid is the naturally present in the certain bacteria. Even it is present in the E. coli. So E. coli plasmid was the first to discovered, and it is used, and it is widely being used. Okay, it is modified nowadays, but it is the main was the E. coli plasmid. So these plasmid vectors, if you zoom out this plasmid, there are certain genes present in that, and that genes helps us to carry the new gene from one location to another. So suppose if I wanted to make a insulin protein, so I have to isolate the insulin gene from the human cells. So I have done the isolation of insulin gene from the human cell. Now I have to transfer that gene to the host cell for the multiplication or for the production of the insulin. So host cells are some mostly the bacteria. So how I can do that? So I will just cut the insulin gene from the human cell, isolate it, and next step is uh, I have to cut the vector, and I have to add that insert that insulin gene into the vector using the enzymes which we have already discussed, that is restriction, digestion, and the ligation. Okay, and after that the recombinant DNA is ready, and that recombinant DNA will be used for the production of insulin in a larger scale. Okay, so they have certain, this plasmid vector has certain, you know uh, genes, important genes that helps in the transporting of the new gene to the new location. Just like your vehicle has fuel, they have certain, uh, the tires to transport it, okay, and seats to sit comfortably. So just like that, the plasmid also has certain genes that helps us to transfer the new gene to new location. Okay, so there are certain essential features that makes the plasmid as a vector. Okay, first is the small size, as it is present in bacteria. So bacteria is already in a small size, so the plasmid is also very small in size. So this plasmid is easily transferred from one cell to another. Okay, so because of their small size. Next, they have certain genes: replication of origin, incompatibility gene, and the par sequence. So replication of origin, it helps in the replication of the gene. Okay, so if you want to produce certain gene in the more amount, you required a replication, right? So they have the plasmid already have the replication of origin for the replication. Incompatibility gene, okay, and the par sequence. Par sequences helps to distribute this daughter plasmid into a stable new cells. Okay, so these are the certain gene sequence that are help in plasmids. Next feature is absence of transfer or mobilizing gene. So if you see in the genome of certain bacteriophage viruses, they have certain genes that can change the position, that can change the location. Okay, so we don't, so after the changing location, the sequence of the gene can be changed, so we don't want that. So in the plasmid, there is a naturally absence of these genes, so there is no change in the sequence, so plasmid will be as it is. Next, they have the marker genes. So marker genes means what? See, why we use marker? So we use marker to label something, right? To identify it. So the marker genes are used to detect the cells that contains the plasmid. Okay, so how we can do that? So there are certain drug resistant genes which is naturally present in the plasmid, such as ampicillin resistance, tetracycline resistance, kanamycin resistance, streptomycin resistance, chloramphenicol resistance. So these are the certain marker genes which are naturally present in the plasmid vector. So that's why it makes it more easily detectable in the plate. So how this can be done, we will discuss it when we discuss the example of the plasmid vector. This will help in the identification of the recombinant genes. They have the next feature is that they have their unique restriction site. So restriction site is, if you remember, in the restriction enzyme, we they cut the restriction enzyme cut at the specific site, right? So that site is known as the restriction site, also recognition site. So in the plasmid, there are certain restriction sites for different restriction enzymes. So you can use any restriction enzymes to modify your plasmid, okay, which is naturally present in it. So that makes it very, easily modifiable. Next, they have the multi-copy nature. Means, see, you want to produce that genes, right? Produce the multiple copy of the genes in a host cell. So you required the replication of origin as the plasmid already have, and also they have the multi-copy nature. Means they can produce a multi-copy. Means, see, in a human cell, we have a one DNA per cell. Okay, one copy of genome per cell. And if there is a replication in the during the replication during the cell cycle, we have the two copies of the DNA, and then the two copies will be separated in the two daughter cells. Okay, so you normally we have as one genome per cell. But plasmid, what plasmid has the feature that in a one bacteria cells, there you can you there you can see the multiple plasmids in it. Okay, means one bacteria cells can contains 1 to 10 plasmids. Okay, so it becomes, very easily isolable, or you can isolate that plasmid easily. Okay, so then we have the two mode of the replication: stringent mode of replication, relaxed mode of replication. In the stringent mode of replication means what? The plasmid replication depend upon the bacterial replication. So when the bacteria is replicated, then on only then your plasmid will be replicated. But in the relaxed mode, the plasmid will be replicate alone. So it will not depend upon the replication of the bacteria. So this makes the, so there are certain plasmid which is in the relaxed mode of replication. We use the plasmid for the recombinant DNA. So these are the certain common plasmid vectors which we use in that PBR322 POC. These are the main vectors, main plasmids we use for the uh, recombinant DNA. Okay, so we will study those examples in the next lecture. So meanwhile, you can just rewatch the lecture of the enzymes or the introduction to genetic engineering. So you will have the sequence. Means you can understand what are the vectors are. Okay, so that's it for today. In the next lecture, we will discuss the examples of plasmids. In that, we will discuss PBR and the PUC. So thank you.